In this video, I'll review concepts from Chapter 18 of Human Anatomy and Physiology, 11th edition. This chapter focuses on the heart. In particular, we'll be looking at the two major categories of cardiac cells. So first, we'll have cardiac contractile cells. These are also called cardiac muscle cells. And then second, we have cardiac conduction cells, and this includes the pacemaker cells of the heart. So let's start with these contractile cells, the cardiac muscle cells. So just as a review of the structure of these cells, cardiac muscle cells are striated and branched. So we can see here, here's one side, and then it splits into two, or at least two. It branches, just like a tree branches. Within each cell, you're going to find typically one, but every once in a while you'll find two nuclei in a cardiac muscle fiber, and they are connected by these things called intercalated discs. So that's shown here, these dark lines that are crossing at different spots along the fibers. This is actually the joining of two adjacent fibers, and when you look closely at the intercalated discs, you're going to see a couple of major structures. First off, we're going to see gap junctions. These are basically pores that allow for things like ions to move directly from one cell to the adjacent cell. This means that when one cardiac muscle cell is depolarized and has an action potential, that's going to immediately travel because that's all based on ions flowing. And those ions are going to immediately flow into the adjacent cell and allow it to experience a nearly simultaneous action potential. Additionally, desmosomes are going to keep those adjacent cells from pulling apart from each other. Remember, these cells are going to really massively contract whenever they're depolarized. And when they contract, these desmosomes are going to help to hold the adjacent cells together. When you look within the cardiac muscle fiber, what you're going to notice is it has a lot of mitochondria. So this is going to be far more mitochondria than what you would find in skeletal muscle. That's because these cardiac muscle cells need to be able to keep going. Basically, they have to keep going your entire life. And so there needs to be a constant, reliable source of energy, and that requires a lot of mitochondria. So these cells are incredibly packed with mitochondria, along with all of these myofibrils that are the contractile units. Again, I'll notice it's so much that about a third of that cardiac muscle fiber is filled with mitochondria. It's a lot of mitochondria providing essential energy for the heart. More comparisons now between cardiac and skeletal muscle. We already have seen cardiac muscle has a lot more mitochondria than skeletal muscle. But now let's look at four similarities. So action potential, depolarization, in both of these muscle fiber types relies on voltage-gated sodium channels. In both fiber types, action potential is going to travel down T-tubules from the surface to the core of the muscle. Action potential triggers calcium release from the sarcoplasmic reticulum in both of these muscle types. And finally, excitation-contraction coupling is going to involve calcium binding to troponin, as well as sliding filaments. If any of these things are not ringing a bell for you, I would recommend going back to any notes you might have or going to previous chapters in the textbook that review these concepts in skeletal muscle. You would have learned about this in an Anatomy and Physiology 1 course. But these are some similarities between cardiac and skeletal muscle. Now some differences. First off, cardiac muscle cells, some of them actually have autorhythmicity. And we'll talk a little bit more about that coming up, but the name sort of is descriptive here. So auto means self, rhythmicity is rhythmic. So they're cells that have their own rhythm. This might make sense. Your heart is constantly beating. That beat has a rhythm. The rhythm actually comes from these autorhythmic cells in cardiac muscle. This does not occur in skeletal muscle. So skeletal muscle fibers, they require nervous system stimulation in order to contract. Next difference, stimulation travels between fibers through gap junctions in cardiac muscle. This is what I showed you a few slides ago. So at that intercalated disc, we have gap junctions that allow for uh, direct signals between adjacent cells. This does not occur in skeletal muscle. Contractions can summate in skeletal muscle, but not in cardiac muscle. Really, in cardiac muscle, we really want to be able to control the contraction. We want it to be exactly the size it is supposed to be in that cardiac muscle, and it just works better to have contractions that do not summate in cardiac muscle. And the cardiac muscle, finally, is going to have much longer action potential and contraction phase than skeletal muscle. So there's just some differences between cardiac and skeletal muscle, in addition to the fact that cardiac has far more mitochondria than skeletal. If you want to look further at this, this is the table from your book that goes through a lot of these different similarities and differences between skeletal and cardiac muscle. So that was a look at the contractile cells of the heart. Now let's look at the conducting cells. These are non-contractile, so they don't actually contract, but they do distribute electrical signals throughout the heart, hence they conduct. They can be called conducting or conduction cells, and you can see them here in yellow moving through this pink myocardium. So the yellow is the pathways of the cardiac conducting cells. These cells are going to coordinate electrical signals. Additionally, 1% of those conducting cells are actually going to be a type of cell called a pacemaker cell. So they have pacemaker potentials. These are the cells I think I mentioned briefly previously called autorhythmic cells. So these 1% of conducting cells have something called autorhythmicity. They develop their own rhythm. So here is a tracing from that. We're looking at changing membrane potential over time. And what you see here is something called a pacemaker potential. So that can be defined, and it is in your textbook, as an unstable resting membrane potential that rhythmically triggers action potentials without any external stimulation. So within that 1% of conducting cells that are the pacemaker cells, this is just happening. It doesn't require any external stimulation. And so here we see that slow depolarization, that's the pacemaker potential, leading to the rapid depolarization and repolarization of the action potential. So this pacemaker potential is allowing the heart to beat without external stimulation. We have three major stages of membrane potential cycle in those pacemaker cells. First off, we have the pacemaker potential itself, number one. This is caused by a slow leak of sodium into the cells. So here we have sodium slowly leaking into those conducting cells. Once it reaches the threshold potential for this action potential, we're going to hit the next stage of that cycle, which is depolarization. And note, this is very different than what we saw in neurons or in skeletal muscle, because here the depolarization is actually caused by an influx of calcium into the cells. So one was a leak of sodium into the cell. Two, the depolarization is a flood of calcium into that conducting cell. And then stage three is repolarization. And in this case, it looks a lot like what you saw in neurons and in skeletal muscle fibers. It's caused when the potassium channels open and potassium starts to flow out of the cell. Additionally, at that time, those calcium channels are going to close. That just allows this to be pretty rapid. So again, we have pacemaker potential into depolarization, and then finally, stage three is repolarization. Compare that now to what happens in a cardiac contractile cell. So this is also going to look a little bit different than what we saw previously in skeletal muscle and what I just showed you in the conducting cells. So in these cardiac muscle cells, what we're going to see is first depolarization is going to occur. So that's stage 1, and this is the blue line. So you can see that's first stage is extremely rapid. So in this case, we're looking at time in milliseconds of about, say about 250 milliseconds. But almost instantaneously, we see that depolarization. And in this case, it's caused by an influx of sodium into the cell. Then the sodium channels close, but calcium channels open. And those calcium channels opening causes an influx of calcium. And so in this case, in these cardiac muscle cells, that influx of calcium is not going to depolarize, it's just going to maintain a plateau of depolarization. So this is phase 2 of an action potential in a cardiac muscle cell. We had depolarization, now we have plateau phase, and then finally the calcium channels are going to close, the potassium channels are going to open, potassium flows out, and you get that repolarization. So these cardiac muscle cells, they are going to have membrane potentials that look a lot different than the cardiac pacemaker cells shown here. So that's pacemaker. You see pacemaker, depolarization, repolarization. And in the cardiac muscle cells, you have depolarization caused by sodium, plateau caused by calcium, and repolarization caused by potassium. I'll leave you with a question. Influx of which ion is going to cause depolarization of cardiac pacemaker cells during an action potential?